Hello students, today let us begin with a new topic, gametogenesis. Before we go into the details, let us first break the word so it becomes easy to understand. The term gametogenesis is made up of two parts, gameto and genesis. Gameto means gamete, that is the sex cells. We know that the sex cells are sperm in males and ova or egg in females. And genesis, which means formation or origin. So gametogenesis literally means formation of gametes. In simple words, it is a process by which sperms and eggs are produced in living organisms. Gametogenesis in males is known as spermatogenesis since it involves the formation of sperms. And gametogenesis in females is known as oogenesis since it involves the formation of eggs or ova. Gametogenesis involves a special kind of cell division known as meiosis. As we all know that the gametes must have half the number of chromosomes. The gametes are haploid when compared to the normal body cells. Because during fertilization, when sperm and egg fuse together, the chromosome number is restored to normal. That is why the gametes are produced as haploid, so that the number of chromosomes for a particular species will be constant from generation to generation. So gametogenesis always involves meiosis. Meiosis is a kind of cell division that reduces the number of chromosomes into half. Now let us walk through meiosis in brief. It starts with a reproductive cell or germ cell which will be diploid, and it undergoes two successive meiotic divisions. Meiosis consists of two divisions, meiosis 1 and meiosis 2. Meiosis 1 is the reduction division which reduces the number of chromosomes into half. So at the end of meiosis 1, we'll get two haploid daughter cells. And meiosis 2 is equational division similar to mitosis. So these daughter cells enter meiotic division 2, and at the end of meiosis 2, we'll get four haploid daughter cells or four haploid gametes. And these gametes are genetically unique or genetically different because of this particular cell division, which involves two key processes or two key steps like crossing over. Crossing over occurs in the prophase 1 of meiosis 1, and then independent assortment or random assortment of chromosomes which occurs in the metaphase 1 of meiosis 1. So there will be different combinations of chromosomes are possible in the resulting gametes. So the gametes are genetically different from the others and from the parental cell. This is the reason behind variation. That is why siblings look different even though they come from the same parents. Now let us discuss spermatogenesis in detail. The gametogenesis in males is known as spermatogenesis since it involves the formation of sperms, and it occurs in the seminiferous tubules of testis. So the location of spermatogenesis is seminiferous tubules of testis. Okay, now we can see the cells involved in spermatogenesis. Different types of cells are involved in spermatogenesis. We'll start with the starting cell, that is primordial germ cell. Primordial germ cells or PGCS. These are the starting cells which we can see in the sex cord of a newborn baby until it is developed into seminiferous tubules. Okay. Primordial germ cells or PGCS undergo repeated mitotic division to form spermatogonial stem cells. These are the stem cells of spermatogonia. And these cells again undergo division to form spermatogonia. Spermatogonia are the actual sperm mother cells. These are the cells from which the sperms are produced. And these cells enter meiotic divisions. These cells enter meiosis to form sperms. So primordial germ cells or PGCS, spermatogonial stem cells, then spermatogonia. Spermatogonia are the sperm mother cells, and then sperms. As I told you earlier, the primordial germ cells are seen in the sex codes, of fetal and neonatal life. And these sex codes are solid codes of cells. They actually represent the immature seminiferous tubules. Understood? Sex codes are immature seminiferous tubules. When the baby reaches puberty, the sex codes transform into seminiferous tubules. The sex codes transform into mature seminiferous tubules. So in the first diagram, you can see the primordial germ cells. Primordial germ cells are supporting cells. Supporting cells, I mean the precursors of Sertoli cells. in the sex code of a newborn baby. Okay, that is given in this diagram. In the second diagram, you can see the sectional view of seminiferous tubule at puberty. In the puberty stage, these sex codes transform into seminiferous tubules. The sex codes mature into seminiferous tubules. And in this stage, the primordial germ cells are no longer seen. We cannot see primordial germ cells in the mature seminiferous tubule. When you reach puberty, we cannot see the primordial germ cells. Instead of that, we can see spermatogonia, spermatogonial stem cells, and spermatogonia of different types. We have already seen that spermatogonia are the mother cells of sperms. The sperms are produced from spermatogonia. So we can say that the primordial germ cells are the precursors of spermatogonia, or primordial germ cells are the precursors of sperms, because sperms are produced from spermatogonial cells. Alright, and these primordial germ cells are seen in the fetal and neonatal life. When the organism reaches puberty, you cannot see primordial germ cells anymore. These cells will be replaced by spermatogonial cells in the mature seminiferous tubule. Okay, now we understood that spermatogenesis in males begins only at puberty as per the maturation of testes and seminiferous tubules, and it continues throughout the life. In the case of males, spermatogenesis will continue throughout their life. There is no time limit. And the location of spermatogenesis is testis. In the testis, inside the seminiferous tubules. Okay, and spermatogenesis duration, I mean the division and differentiation of one spermatogonial cell into the gametes, it will take 74 days. The duration of spermatogenesis is 74 days. Likewise, likewise males have millions of spermatogonial cells in the seminiferous tubules. Around 300 to 600 millions of spermatogonial cells are there in the seminiferous tubules, and around 300 million sperms are produced per day. Okay, now let us explore the process. Okay guys, imagine this as a flow from left to right in the seminiferous tubule, starting from stem cells and ending as fully formed sperms. On the extreme left, you can see the spermatogonial stem cells. These are the original parent cells. These are the original parent cells from which the spermatogonia are formed. Spermatogonia and sperms are formed. Okay, and these cells are diploid. I mean, they have 23 pairs of chromosomes. They have 46 chromosomes, half from maternal side and half from paternal side. So they are diploid, and they act like the stalk. They are the stalk that keeps on renewing. These cells will divide mitotically and renew themselves. Okay, from these stem cells we move into type A spermatogonia. The spermatogonial stem cells undergo repeated mitotic division and then differentiate into spermatogonial cells, the actual mother cells of sperms. Alright, and the spermatogonia are named as type A. They are type A spermatogonia, or they are named as type A dark spermatogonia because they have dark nucleus. The nucleus is darker. Understood? And they are also diploid. The type A spermatogonial cells divide mitotically to form type A dark, again AD cells, and then AP cells. AP cells means type A pale spermatogonial cells. The type A pale spermatogonial cells are so called because the nucleus is lighter when compared to type A dark spermatogonial cells. Their nucleus is light. They have less dense nucleus when compared to the type A dark spermatogonial cells. Type A dark spermatogonial cells have darker or more dense nucleus. Alright. And type A dark spermatogonial cells, AD cells and AP cells divide mitotically. They renew themselves, and from AP cells, type A pale cells, type B spermatogonial cells are formed. Type B spermatogonial cells. So what I told you, spermatogonial stem cells, from spermatogonial stem cells to type A spermatogonia, type A dark spermatogonia, and from type A dark spermatogonia to type A dark again, and then type A pale, AD and AP cells, and from type A pale spermatogonia to type B spermatogonia. That is, type B spermatogonia are formed from type A. What kind of type A? Type A pale spermatogonia. And type B spermatogonia then differentiate into primary spermatocyte. and these are the last diploid cells in the seminiferous tubules. And the primary spermatocyte then enter the growth phase of cell cycle. You know the different phases of cell cycle like G1 phase, S phase, G2 phase, and then the division phase or M phase. Okay, the primary spermatocyte enters the growth phase. It grows, its DNA replicates. I mean DNA synthesis occurs and it prepares itself for cell division. Then it enters the first meiotic division. It completes the first meiotic division to form two haploid daughter cells. You know that the first meiotic division is the reduction division in which the number of chromosomes are reduced into half. So we'll get two haploid daughter cells. These haploid daughter cells are known as secondary spermatocytes. Okay, primary spermatocyte and then secondary spermatocytes. Secondary spermatocytes are haploid. These cells then enter the second meiotic division. They complete the second meiotic division to form four haploid daughter cells. You know that second meiotic division is similar to mitosis. Okay, as a result of that, four haploid daughter cells are formed. They are known as spermatids. They are spermatids. We'll get four spermatids. And from these spermatids, sperms are formed by the process of spermiogenesis. These spermatids are morphologically changed into sperms. So at the end of spermatogenesis, four haploid sperms are formed. Okay, now you should remember that inside the seminiferous tubules, there are different types of spermatogonial cells like type A and type B. Among these, type B spermatogonium forms the primary spermatocyte. Type B spermatogonium mitotically divides and then differentiate into primary spermatocyte and then to secondary spermatocyte and then to spermatids and sperms. Alright. Okay, this is the generalized diagram showing spermatogenesis. It begins at puberty and it consists of four stages. The first stage or first phase is multiplication phase, in which the spermatogonial cells undergo repeated mitotic division and then differentiate into primary spermatocytes. The second phase is growth phase. In this phase, the primary spermatocyte grows in its size. The primary spermatocytes enter the growth phase of cell cycle and prepare themselves for meiotic division. The third phase is maturation phase. In this phase, the primary spermatocytes complete the meiotic divisions, meiosis 1 and meiosis 2. At first, the primary spermatocytes undergo the first meiotic division to form two haploid cells. They are known as secondary spermatocytes, as we have seen already. Okay, then the secondary spermatocytes complete the second meiotic division to form four haploid daughter cells or four haploid spermatids. Okay, and the last phase is spermiogenesis. In this phase, the spermatids differentiate into sperms. The spermatids undergo morphological changes to form sperms or spermatozoa. Okay, these are the four steps in spermatogenesis: multiplication phase, growth phase, maturation phase, and then spermiogenesis. Okay, and we will discuss the spermiogenesis process in the next class.